Hopefully everybody has done the warm up. I have gone in and graded the ones that had been turned in a little while ago and also corrected. A lot of people misspelled acetylcholine. Again, one of the stupid things about these online systems are they are so literal. So I actually gave it a few possible misspellings that I thought people might make, but people came up with all sorts of other new misspellings that I'd never imagined existed. So I went in and manually corrected those. Again, so I'm still trying to figure out the best way to do this online testing, quizzing stuff, and what are the strengths and weaknesses and quirks and all that. So let's go into the warm-up and preview. So let's take a look. Here was question 1. And here you have a pretty nice picture of, let me get the little drawing, some part of the nervous system. There's like neurons, there's ganglia. So, and it says, what part of the nervous system is this from? So what did people say?
Sympathetic.
Yeah, it's totally sympathetic. So that was the answer there. Why? How do you know? And again, on a normal warm-up, I would just have a second part saying, how did you know? But it's harder to do on this. So how did you know?
Thoracic lumbar origin.
Yeah, so one is it's originating from the thoracic and lumbar region of the spinal cord. So that thoracolumbar outflow. You know, if it was going to be parasympathetic, it would be originating from cranial nerves and from the sacral region. But it's from the thoracic and lumbar region. So that's a giveaway. What's another giveaway?
Ganglia chain.
The sympathetic chain, this sympathetic chain or sympathetic trunk, these ganglia going up and down near the spinal cord, that's something that is a hallmark of the sympathetic division. If this was parasympathetic, there would just be a big long neuron going almost all the way to the target, and then an itsy bitsy second neuron right at the target, those we call the terminal ganglia or intramural ganglia. So, but in this case, and a thing that goes along with the fact that the ganglia are near the spinal cord is that you have a short first one. You know, this first neuron that leaves the spinal cord to the ganglia is short, and the second neuron that continues going all the way to the target is long. So short first, second long second, that's another hallmark here. There's one other thing here that's probably a little trickier to notice, but if you kind of take a look here, it's like, oh, it's also one of the targets is the adrenal medulla. So that is another giveaway that this is better be the sympathetic. The sympathetic system is the one that triggers the adrenal medulla to release adrenaline into the system. So lots and lots of different clues to tell you this is the sympathetic nervous system that we're seeing. And actually, this is another one here. I don't know if you remember, I talked about one of the ways that they are, we talked about sympathetic, parasympathetic in general being antagonistic, pushing and pulling on each other, but sweat glands are actually purely sympathetic. So if you're activating a sweat gland, you're probably looking at the sympathetic nervous system as well. So lots and lots of clues that this was sympathetic.
The first neuron looks like only one neuron. The long one, it looks like parasympathetic, the first one in the beginning.
Wait, we'll say it again. Say that one more time.
The top one, the one is connecting with the eye, it looks like one neuron. I didn't see two neurons.
Oh, the first neuron is the red one that synapses in the ganglion. And the second neuron starts in the ganglion and continues on to the eye. Right? There's this red neuron is the first neuron and the black neuron is the second neuron. And we said sometimes you just go into the sympathetic chain, synapse there, and the second neuron then gets onto a spinal nerve, continues on. These are representing the splanchnic nerves where you're going through the chain, synapsing in one of these collateral ganglia, and then the second neuron continues on to the target. But in both cases, and these terminal ganglia are also near the spine. So this is a short first neuron, and then the black one is the long second neuron. Does that make it clearer?
Yes, thank you.
Okay. All right, so let's clear that stuff. Next, so postganglionic sympathetic motor neurons. So sympathetic, what are they going to release?
Epinephrine.
Yeah, these are going to be epi, or the thing that's more generally used as a neurotransmitter is what?
Norepinephrine.
Norepinephrine. So norepinephrine. Epinephrine tends to be more of a hormone. It's released by the adrenal gland in the bloodstream and goes everywhere. The same receptors that will bind norepinephrine will often bind epinephrine. So they both bind the same receptors, but typically norepinephrine is found in neural synapses, whereas epinephrine is dumped into the bloodstream as a hormone. As opposed to that, parasympathetic releases what typically?
Acetylcholine.
Yeah, acetylcholine. What other motor division released acetylcholine at their targets?
Somatic motor.
Exactly. Somatic motor also released acetylcholine, but the difference there was one was the targets were skeletal muscle, and also that the skeletal muscle targets had these nicotinic cholinergic receptors, which are always excitatory. Whereas when you're in the autonomic system and you release acetylcholine at your target, sometimes you excite the target, sometimes you inhibit the target. Depends on the particular situation. All right.
Didn't you also say that sweat glands secrete acetylcholine?
Yeah. So that's why I said typically. So in general, sympathetic releases norepinephrine or epinephrine, but there are some exceptions, but they're not the rule. All right, question 3. The white ramus communicans and the gray ramus communicans allow motor fibers to basically get out into the sympathetic ganglia and then have the second neuron join and then continue traveling along the spinal nerve. Is true. We spent quite a bit of time kind of talking about how the white ramus is the off-ramp to get into the ganglion and the gray ramus is the on-ramp to get back onto the spinal nerve. The plural, I realized like if I was going to say white and gray ramus, I would have had to use this, rami would be the plural of ramus, and then you'd say communicantes. And so rather than saying white and rami communicantes when we hadn't actually used that terminology and you wonder what the heck is he talking about, I just wrote it out longer. The white ramus communicans and the gray ramus communicans. But if you see like communicantes, it's like this plural thing going on. Then this next part, name at least two anatomical differences between sympathetic and parasympathetic. So one thing, anatomical. That means like things that are about anatomy, study of structure. So you wouldn't want to say, sympathetic is, excites your for the fight or flight. That's not structure. So anatomical would be things like, sympathetic. We talked about sympathetic was that thoracolumbar outflow, versus parasympathetic was a craniosacral. And we talked about how the ganglia are near the spine, or near the spine when you're in sympathetic, but are close to the target organs in parasympathetic. We talked about how sympathetic has a short first neuron, a long second neuron, whereas parasympathetic has a long first neuron, a short second neuron. So all those kind of things, the things we already kind of talked about when we did question 1. Clear drawings. And then here we go. So now we're talking about the somatic motor division. So how many neurons does it take to get from the CNS to the target for somatic pathways?
1.
One neuron that has its cell body in the CNS, has its axon, goes all the way, and synapses at the skeletal muscle. So the answer here was 1. Again, like I had it so it could recognize 1, it could recognize spelling out 1, but if somebody wrote like 1 neuron, it's like, I don't know what it's talking about. So I have to go in and manually do it. I'm figuring it out. Target for the somatic motor neuron is what kind of a muscle cell?
Skeletal muscle.
It's going to be skeletal muscle. So we could call it striated muscle or even voluntary muscle or something. Motor neurons release, we talked about that, acetylcholine. So any questions about our virtual warm-up? Okay. I'm going to...
I do have a question. This is unrelated to the warm-up before you start in on the lecture. I just was curious if there was a way to have our next quiz on Tuesday or Thursday of next week, just because it's such short notice, so we have more time than a day to study. Just wanted to run that by just to see if there was a chance for that.
Yeah, no, it would be way less stressful for me.
I'm not sure if other people want that too. I just know for me, I don't have that much time to study tomorrow, and I didn't know that that was coming up.
However, it is easier for everyone.
I'm fine with waiting until next week.
I'm good with next week.
Yeah.
That's going to be 4-7. Yeah, no, that, again, I'm kind of making this up as I go. So any feedback from you guys is helpful.
Thank you. Appreciate that a lot.
Sure. Okay, so yeah, so everybody has that, and luckily this is what's recorded on the it's going to be up on the YouTube. So quiz. is going to be on Tuesday, 4/7, and exactly what it's going to look like, we'll figure out before then. All right. So yeah, thank you for that. All right. So now let's go. All right. So we need to do the sensory division right now. It's going to be kind of the last part of the nervous system. We've already talked a bunch about the eye. I'm really, really freaking glad we did at least a sheep eye dissection when we had a chance. So you had at least, yeah, you got to think something like that. Like, unless you actually are there and see that beautiful tapetum lucidum behind the retina on a sheep eye that looks like some beautiful abalone, it's like, you could read like, oh, and the sheep eye has an extra layer. It's called the tapetum lucidum, which is iridescent behind the retina. It's like, okay, whatever, factoid. But like when you see it, it's like beautiful. So that's the kind of thing I'm sad that you guys are gonna be kind of missing. And at the same time, again, when we get to the lab part today, I'll show you some of the stuff that I found that we can use to try to make up for at least some of that experience. But so sensory, we're gonna kind of introduce the senses. And then we're gonna kind of focus on mostly, your five senses. So we're gonna do the general senses like touch, touch and pain and temperature. We're gonna finish the eye. We're gonna do, olfaction, smell, and gustation, which is taste. I can spell. And then we're gonna get into the ear. Again, the ear does a lot of stuff. The ear is gonna be hearing, but it's also gonna be a bunch of other equilibrium senses. So that's our plan. Over the next hour, we're going to finish up the nervous system by covering the senses. And then for our lab time, we can get deeper into both the ear, taste and olfaction, the eye, general senses, all have cool stuff to look at under the microscope. Again, I'll show you pictures of what it looks like under the microscope, but you still are going to be responsible for recognizing the histology through pictures versus looking through a microscope. I will also introduce a couple of other resources. So once we get to lab, we'll get more into detail of this stuff, as well as talk about some of my ideas of how to approach lab. Okay, dokie. So stimulus is just there's some thing out in the universe, versus perception. This is like what you experience. So like sometimes it's a clear connection, like, you turn on the lights and you see, oh, the room's lit up. Or, don't know, a bird lands on your shoulder and you can feel like its talons digging into your shoulder. There's a lot of stimuli that are happening, you know, there are radio waves coming through the air, and you don't have any idea that there are radio waves. In order for this to happen, there's two steps. One is we need to have some way to have our nervous system. Our nervous system is what's kind of mediating all of our experience of the world. So we have to have some way to convert this stimulus into something that our nervous system is going to be able to process. And the main thing our nervous system processes are electrical signals coming down neurons. So at the core of the sensory division here is going to be this concept of sensory transduction. Maybe I should probably write it neater because it's actually super important concept. This is basically where you take the stimulus, whatever it is, and you convert it into an electrical signal that then the nervous system can process. So this is basically convert stimulus. You know, and the stimulus could be anything. The stimulus could be light. The stimulus could be bending of your skin for touch. It could be the presence of sugar on your tongue for tasting sweet. So the stimulus is just something out there. You have to convert it into an electrical signal. You know, for the nervous system to process. The actual little part of your body that does this job, this is performed by receptors. So you have receptors like photoreceptors that detect light. They convert light energy into electrical signals for your nervous system to process. You have mechanoreceptors that sense the bending and deformation of your skin and convert it into electrical signals that go into, you know, your somatosensory cortex. You've got all sorts of different receptors that we're going to be looking at that are doing sensory transduction, right? So if you don't have a receptor to do this, then you do not have any way of even knowing the stimulus exists, right? So if, like the example I gave of radio waves, you know, there are lots of radio waves cutting through the room. You know, my room, your room out there, you know, she wouldn't be hearing me if you didn't have your Wi-Fi going. The Wi-Fi is pulsing. All these electromagnetic things are pulsing around everywhere. Unless you have some instrumentation that can actually detect it, you know, you don't have the receptors to detect it and notice that it's happening. So you also have receptors that have limited ranges, even if for a sense that they do detect or stimulus they do detect. You know, for hearing, we saw, you know, your ears can detect air vibration as sound, but if it's too high frequency, you can't hear it anymore. And in fact, even where that cutoff was different for different people. If you were a cat, you could hear things that were, you know, 3-4 times the frequency that a human can. You know, other things, if you were a bat, you could hear frequencies up above 100,000 hertz, whereas, you know, I can't even hear like 15,000 hertz. So, you know, what you actually can detect is going to depend on the limits of your receptors. it's kind of a fascinating thing. I mean, I'm sure most people have seen the movie The Matrix, where they just are kind of like your brain is creating your reality based on the inputs that you're being given. But that is kind of what's happening. You've got receptors on the surface of your body. You've got your retina on the back of your eyeball. You've got your smell and taste receptors inside your nose and inside your mouth. You've got touch receptors on your skin. You've got your eardrums moving back and forth, like right on your ear. And so basically there's this interface between the universe and the surface of your body. The receptors pick up information about what's happening at that interface, and your brain creates this virtual reality that you project outside of yourself as the world you're living in and everything that's out there. But if you really think about how you create that, it's from this pretty limited set of data that is being, picked up by these receptors doing sensory transduction on the surface of your body. So it's kind of trippy. In physiology, we spend more time looking at kind of how this works, how it doesn't work. Well, but obviously you've probably realized it's not that easy, not that hard to create different kinds of illusions and things like that, because we're doing our best to create a model of what this information means, but it's easy to fool it as well. So let's continue on. Page 2. So we talked about receptors. We can talk about receptors. Again, this class, even though we are now online, we are still going to be just categorizing things, structural classifications, functional classifications. That part doesn't end. So there's lots of different ways we can characterize receptors. One way is by where the stimulus is that they're responding to. So there are like exteroreceptors that are detecting things outside of your body. This would be like your 5 senses, vision and smell and hearing, where the stimuli originate outside of your body. However, there's a lot of receptors that are monitoring processes inside your body. Like how stretched out is your stomach? What's the pressure of your blood right now? What's the sugar levels in your body? How much carbon dioxide is building up right now? So there's a lot of information your body needs to maintain homeostasis and keep all your processes going. So we have what we call interoreceptors. Sometimes they're called visceroreceptors. Viscera is just like organs. So again, these are from stimuli outside of the body. The visceral or interoreceptors are usually not perceived as a kind of conscious perception. Your brain is getting information about sugar levels in your blood. It's getting information about how stretched out your blood vessels are at the moment. It's getting information about all sorts of internal processes in your body. And it is actually using those for these reflexes to control things like, just for, the basic idea we talked about reflexes. It's being like a stimulus, integration, and a response. Some of these are very familiar to you. You like hit somebody's knee with like a little reflex hammer, and it goes into their brain and actually just mainly in the spinal cord, and then this response is they kick out their leg. Right, so you're all used to that as a reflex. There's a stimulus, you hit their knee with a hammer. It's integrated in the CNS, and the response is you kick out. There are a lot of reflexes that you are not consciously aware of that are like using these. You know, you could have a visceral reflex where the stimulus is kind of your stretch of your blood vessels. As the blood vessels are getting like more flaccid, they're not as stretched, your nervous system is getting that information and is going to respond and is going to do things to try to crank up your blood pressure, maybe speed up your heart or this or that. There's lots of different ways your body cranks up your blood pressure. It's going to talk to your kidneys and say, retain more water to bring up your blood volume. So there are lots of reflexes that are happening that you are not aware of that are maintaining your homeostasis. Baroreceptor reflex is one. Like again, if you have these little blood pressure receptors that are saying your blood pressure is dropping, you know, within a few moments, you're going to have all sorts of responses happening that you're not aware of, but that are really essential. Because like when you're lying down and you stand up, if you don't have those reflexes happening, you're gonna black out and face plant. So yeah, so think about when you think about receptors, it's not just about the five senses. It's also about lots of things inside your body. Again, the reflexes that involve these interoreceptors are usually more likely to involve the autonomic motor system to respond, whereas the exteroreceptors are probably more likely to be using somatic motor. You're having some kind of physical response. Okay, other classifications of receptors. This can be classified by where the stimulus is from. We can also classify them by what kind of stimuli they respond to. So classification by stimulus type. So what are some of the kinds of stimuli we can respond to?
Touch.
Touch. So touch is when you're actually mechanically kind of stretching and deforming your skin. It's going to be an example of what we call mechanoreception. So we have mechanoreceptors. Mechanoreceptors would be for touch or pressure. They also are used for lots of other things. They're actually what help you determine blood pressure by looking at how stretched out your blood vessels are. It's maybe not totally obvious at first, but hearing even. Hearing is mediated by mechanoreceptors because sound waves are just vibrations of the air, basically these compression waves through the air, which then hit your eardrum, and your eardrum is basically vibrating, moving back and forth. And that vibration is ultimately going to get transmitted to little mechanoreceptors, as we're going to see by the end of today. So, oh, hold on, all right, I just saw the chat coming up, but I hadn't been following it, so I now have the chat window open here. Oh my God, there's all sorts of stuff you guys are doing. All right, so mechanoreceptors, again, hearing, we're going to see as a mechanoreceptor because it's actually the movement of your eardrum that has to get converted into an electrical signal. So cool. What other kinds of stimuli can we respond to?
Chemical stimulation.
Totally, chemicals. So chemoreceptors. So, you know, for the five senses, this would be like taste. Taste can detect the presence of sugar dissolved in your saliva and send a message to your brain saying there's the presence of this particular chemical. Smell. Smell is another example of chemoreception. You know, the experience of one smell or another is just the presence or absence of a particular type of molecule floating around in the air. So chemoreceptors. You know, internally, your body is using chemoreceptors to know what kind of food is inside your digestive tract. What kind of enzymes do we need to break it down? Are we done breaking it down yet? Is the main food molecules gone and the breakdown products show up? The chemoreceptors are going to be looking at sugar levels in your bloodstream or carbon dioxide levels in your body to make sure you're breathing enough. So there's lots of chemoreceptors on this internal side as well. What other kind of receptors do we get?
Light receptors.
I'm gonna say Nikki beat you to it. Light. So light, we're gonna call photoreceptors. And those we're gonna look at in more detail today, the rods and cones in the retina of your eyeball. But wait, there's more. What's another thing we can detect?
Touch.
So touch, so Chris, so touch would come under mechanoreceptors. Another one that we just saw up there was nociception. So these are the things that we perceive as pain. So pain is the conscious experience of activation of the nociceptors. What is the stimulus though? What is the actual stimulus that, you know, like the presence of sugar at a taste bud is the actual stimulus that you perceive as sweetness. What is the actual stimulus that you perceive as pain?
Tissue damage?
Yeah, it's going to be like tissue damage. Tissue damage or impending tissue damage. And most receptors, actually, if you drive them hard enough outside their ranges, will start to be perceived as pain. You know, if you're just pushing on something too hard, you know, the system's going to start saying like, whoa, we're in danger of hurting something. Then there's one last kind of receptor. We've got mechano, chemo, photo, noci, and what's another one?
Proprioceptors.
Oh, so I am going to get to the proprioreceptors in a moment. Let's talk about thermoreceptors first, which are detecting temperature, and then proprioreceptors or proprioceptors. And as you can probably tell, I'm being a little fast and loose with whether it's a receptor or a receptor. I'm not exactly sure sometimes what, like, you know, thermoreceptor versus nociceptor. If you, on a test, you wrote proprioreceptor or a proprioceptor, it's both good. Don't stress about, but what's the, you know, in this case, I've never kind of quite figured out the rhyme or reason why sometimes they write out the full word receptor in the compound word and sometimes they shorten it and just make it a proprioceptor. So I'm not going to hold you accountable at all, whether you like add in that RE or not. I don't even keep track of it myself. So proprioception, let's talk about this. I'm going to get a new page. This is basically giving you information about the status of your physical body. It's, you know, the reason why I like to, it's mainly these mechanoreceptors that are actually giving you this information. There are So examples of these that you should know. There is the muscle spindles. That detect the stretch of the muscle. So you have information coming to your brain about how stretched out a muscle is. They're the Golgi tendon organs. These are detecting the tension. So we have something that looks at how stretched the muscle is. We have another thing that tells us how much force is this tendon pulling on its attachment. 3, another one is the joint angle receptors, telling you how a particular joint is situated. And these are basically little stretch receptors. So like, let's say here is a classic synovial joint. And here is your joint capsule. This should look totally familiar. Then what this actually is just like little stretch receptors in the joint capsule. So if you bent the joint, let's say you were flexing this, these stretch receptors would get more stretched, these would get less stretched. So your brain could figure out that you were moving the joint this way. If you were moving the joint this way, this would get more stretched, this would get less stretched. So your body, again, your brain would have information to know what's the joint doing, what's the angle of the joint right now. So joint angle receptor, it sounds kind of fancy, but it's basically little stretch receptors that are in the joint capsule that you can use to then figure out what is the current position of this joint. So, you know, like all this information is going like up into your cerebellum to help your brain kind of know, like, I know what my motor plan is, what is my body actually doing, what is the current position and situation and everything. So this is like important information for making, you know, your body actually move the way you'd want it to. It's also just really important for you to feel like you're in your body. When there's problems with this, people get really weird experiences of like not being in their body. So those, in fact, let me double check my notes that I did not forget anything. Nope, I didn't. All right, so now we've kind of finished the kind of introduction to all the different sensory stuff. And again, remember this idea of sensory transduction being at the core of like, how do we use a receptor to take the stimulus, whatever the stimulus is, and then we have to convert it into an electrical signal for the nervous system to process. So now we're gonna spend the rest of our time, we kind of gave a little nod to the internal or visceral receptors, but we're gonna focus on those outside ones, so your classic 5 senses. So we can talk about the general senses and the special senses. General senses, these are the ones that are just located all over your body. These are things like touch, pain, temperature, pressure. They tend to have very simple receptors. So we will be able to cover the general senses in just a few minutes. It's not going to take us that much time because even though they're all over your body, there's not much to talk about when we talk about what they look like or how they're put together. Special senses, on the other hand, these are all located in the head. And they usually have complex receptors. This is where you have like the eyeball, which we saw already is really complicated. The ear is going to be even more complicated. And taste buds, even though they're pretty simple, they're way, way more complicated than just like a touch receptor or something. So we will spend time finish looking at the eye. We'll look at the ear. We'll look at the taste buds. We'll look a little bit at the olfactory epithelium. And these will be pretty quick. This, fortunately, we're done the most of it. And then this will take kind of the most amount of time to finish up. Again, this is supposed to be an ear. You can put an earring in it, maybe. That looks more like an ear. So let's do the generals. Let's look at the time. We're all good. Let's do the general senses. General senses. Sounds like it should be like command, but general senses. So the receptors for the general senses, some of them are literally just little free, what they call free or naked dendritic endings. So like here's our like unipolar neuron sending information into the dorsal horn of your spinal cord. La, la, la. So this is like my little free, sometimes they call them naked dendritic ending. So this could be a thermoreceptor, hot or cold. It could be a pain receptor. It could be a kind of touch receptor. There's no way to know just from looking at it. What you'd need to do is give it some kind of stimulus and see what it responds to. If you make things cold and all of a sudden you start seeing electrical activity, zzz, then, oh, it must be a cold receptor. Or you start making it hot and it starts going, zzz, sending electrical signals, must be a heat receptor. Or if you start tweaking the tissue around there and something happens. So like, you can't tell, if you look at an eyeball, it's an eye that's sensing light. If you look at a heat receptor, temperature receptor, there's no way you'd have any idea that's even a temperature receptor until you see its actual response to a certain stimulus. A lot of the mechanoreceptors are a little more complicated. They're called encapsulated receptors. So there's some connective tissue capsule around the dendrite endings. This includes like the classic little touch receptor. So this would include, for touch, we had these things called Meissner's corpuscles. Which could also be called a touch corpuscle. For deeper pressure, there's something called a Pacinian corpuscle. You've seen this already in the skin, on the skin models. I introduced them. We're going to look at them again. In fact, maybe I should look at them real quick. If I go and I share. Where's this? The skin's got to be in here. Oh, maybe the skin is not in there? Hold on. My apologies there. I was like pretty sure that it would have to have the skin. Let me look at a different. Let's see. Yeah, I'll come back to it in a few moments. Or, let me go into the martini. Dagnabbit. My apologies here for our slight little. So anatomy, classroom files, anatomy, PowerPoint. Let's do. So here, this thing that they're calling a tactile corpuscle. This is what I was just calling a Meissner corpuscle. They are basically like these little, they're like little tic tacs that are located right up in the papillary, the dermal papilla here. So in the papillary layer of the dermis, and these little dermal papilla are these little tactile corpuscles. When we get to lab time, I'll actually show you what they look like under the microscope. They're actually pretty subtle, but you can see them. Other things that are, this is actually kind of small. This thing called lamellated corpuscle. That's another name for Pacinian corpuscle. Lamellated because there's lots of layers. They look kind of like a big onion. They are usually deeper. They can actually be all the way down in the subcutaneous layer even sometimes. Again, when we get to the lab part of the class, I will show you what these look like under the microscope as well. And when we're taking our break, I will find a better picture of the skin that shows these more clearly. So, doobie-doo. All right, so that was basically general senses. We are not going to really talk much more about them other than they're all over their body. They have simple receptors, and you will get to recognize a couple of them, like especially the encapsulated ones, which you can actually they actually have some form. So like the Meissners or touch corpuscles, or they call them tactile corpuscles, and the Pacinian corpuscles, which are these big things that look kind of like onions. All right, so now we're going to go to the special senses. We're doing all right here. So probably makes sense to just finish up the eye since we've already been talking about the eye. And hopefully you still remember the basics. Whoa. Hold on. Hold on. Sorry about that. All right. Maybe I should. So what is the official word for the eyelids that I'm kind of drawing here?
Palpebrae.
Exactly, the palpebrae. And then we had the little lacrimal puncta that you've seen, you got to see in each other in lab, and with the little lacrimal ducts into the lacrimal sac with the little nasolacrimal duct. So that should all be clear to you, or you should remember. What did we call that little mound, fleshy mound that makes all your kind of eye snot or sleepy duck?
Caruncle. Caruncle.
That's the caruncle. Then we have the actual eye itself. You know, what is the eye? What is the pupil made out of?
The whole.
Right, exactly. That's a trick question. I'm actually really not usually somebody who does trick questions, but that was one. Yeah, it isn't anything. The pupil is just darkness because the iris has a hole in the middle of it, and you're looking into the internal cave of the eyeball where it's dark, so it looks black. Iris, we talked about having, what kind of muscle fibers are in the iris?
Smooth muscle.
Smooth muscle.
Smooth muscle, longitudinal fibers that can make it get wider. That would be dilating. Circular fibers that can constrict to make it get more, get a smaller hole. Which division of the nervous system is going to constrict the pupil?
That's going to be sympathetic.
That's not,
Parasympathetic.
it's going to be parasympathetic.
Parasympathetic.
Yeah, sympathetic, part of that, like fight or flight, like your eyes get really wide. It's like, wide-eyed with fright. You bring in all the light. Whoa, what's going on? So dilating your pupils is actually sympathetic. What is the white part of your eye?
Sclera.
Sclera. And then actually we talked about how there was a layer of a mucous membrane that's actually over the top here where the blood vessels are. What do we call that mucous membrane that actually is over the surface of the eye from the front? Conjunctiva, exactly. All right. So we spent quite a bit of time. What the? Oh, shit. Hold on. All right. So we spent quite a bit of time in here, so I'm not going to redo everything. We had the fibrous tunic, which again was this kind of clear window, the cornea. And then the sclera, which is that kind of tough tendon-like part. What was the next tunic?
Vascular.
The vascular tunic. Sometimes it's called the uvea. That was going to have a few different parts. It actually includes the iris. It includes the ciliary bodies. The ciliary bodies are actually the things that attach onto the lens. And then there's this kind of dark layer all along the inside here. Which is the choroid. So there's somebody who's got their speaker on that is kind of, you probably, if you're not talking, you probably want to mute. We can hear this kind of like different sounds. All right, thank you very much. So choroid layer, and again, you saw that when you did your sheep eye dissection. The choroid layer was that black layer that was right behind the retina. You know, in a human, you do not have that tapetum lucidum, which again looked a lot like that beautiful aurora borealis behind me here. I don't remember how much we covered. Did we talk about how the ciliary bodies are making the aqueous humor, which is getting drained by the canal of Schlemm?
Yes.
Okay. All right, good. So I'm not going to go all into that. Basically, these are making this aqueous humor. Little canal of Schlemm is going to be draining it. The vitreous humor back here. Let me not get sloppy. So the one thing we did not talk about specifically was the retina, which is going to be that neural tunic, the tunic that actually has the ability to do the phototransduction. It's the part that literally converts light into electrical signals for the nervous system to process. So we are going to look at the retina right now, and that's the part we're going to spend a little bit of time being some detail. So this little part that this little chunk, we're going to take this little chunk and I'm going to blow it up. So we're going to be looking at the back of the eyeball, the choroid and the retina, and then the vitreous humor in the front of the eyeball is off to the left here. So those are the back. This is the very back of the eye. This is towards the front of the eye. Choroid. For the retina, there is this layer called the pigmented layer, which I'm drawing in black, and it is black actually. The pigmented layer is where the rods and the cones kind of bury their heads to get nutrition and get replenished and all that. So the pigmented layer, and then you have the rods and the cones kind of bearing their heads. The cones get their name because this is what we call the outer segment, looks kind of cone-like. So cones would look kind of like this. They're these weird cells that are actually specialized, adapted from neuron cells. All of the retina originally started as the optic cup off of the diencephalon during development. You probably remember when if you did that drawing, the human brain drawing coloring book exercise they gave you way back in the day. There was a little optic cups coming off of the diencephalon. Those kind of migrate towards the front and they end up becoming the retina on the back of your eyeball. So cones look more like coney on their outer segment, whereas rods look, these are rods, these are cones. They both do the same thing. What do they do? What's their job? What's the job of a rod or a cone?
Interpret light signals.
Yeah, so they are going to be doing the sensory transduction that is going to convert light energy into an electrical signal. So maybe let me spend a couple of moments looking at rods versus cones. You should be able to know how they're similar and how they're different. The big thing is they are both photoreceptors. So they are both in charge of taking light energy and converting it into an electrical signal. But then there's also quite a few differences. One is the rods are able to detect very dim light. So if it's very low light levels, it's the rods that are letting you see. So these work in dim light. Not dark. No, you'll get it wrong if you say, oh, rods see in the dark. Nothing sees in the dark. Dark means absence of light. If there's no light, you don't have vision. But it does give you the ability to see in very dim light. It might be very, very not much light, but there's a little bit, and the rods are going to be able to detect it and let you know there's something there. Cones work in bright light. Right now, if it's bright, you're using your cones. The rods get totally overwhelmed if it's really bright. So they get what's called bleached out. So if you're in a bright room right now, you're using your cones. You're not using your rods. But if it's really kind of dim light, then the cones don't work anymore. And then you have to use your rods. One of the ways you're using rods versus cones, rods are monochrome. They only detect the presence or absence of light. Usually kind of centered around in the green range, but there's only one type of rod, and it either says there's light or there's no light. So when you're looking at the world just using your rods, you don't get a sense of color. You just get a sense of there's something there or there's not. Cones have color. There are actually three main populations of cones. Like if you think about the response, you have one population of cones that will respond mostly if the light is in the frequencies that we perceive as red. They're kind of the lower frequencies. You have another set of cones that are responding in the frequencies that we perceive as green. They're kind of this mid frequency of visible light. There's going to be the blue cones, and these are going to be the ones that respond maximally at the higher frequencies of electromagnetic radiation that we perceive as like the blue up into the violet visible light. Right, so when you are looking at something, like I'm looking at Jamba's background sky there, and I see like that deep blue up in the sky of his background, you know, that's because the blue cones are getting stimulated. The green and the red ones are not so much, so I know that it's got to be blue. How about the white sky? I don't know if you can see the white clouds in his sky there. What does white mean? How does my eye know that it's white light?
No color being detected.
So
Oh, I think
no,
that's
you're almost there. No color. If
Is
there's
that more rods?
yeah, so if there's no light at all, you'd see black. No light at all would be black. White is when you have all the colors happening at once. And it's a little non-intuitive. You know, when you're in elementary school and you're mixing colors, you know, green you get from yellow and blue, and purple is red and blue, and if you put all the colors together, you get like mud. You've probably, I mean, I don't know if you have, I still remember being a little kid being so freaking disappointed when you mix all the colors together and it's just like icky looking. You think it should be like amazing. But with luminance, if you're mixing up luminance, it's different. They don't mix the same as mixing pigment. So if you mix all the colors together in terms of luminous, in terms of actual light, it actually becomes white. That's why, like, when you take sunlight and put it through a prism, the prism is separating the different colors that are actually all mixed together to make the white light. If people have congenital conditions where they're missing one or another of these populations of cones, then they're going to be colorblind, have one form or another form of color blindness. So if you have like rich colors when you're looking around you, you know you're using your cones, because the rods can't do that for you. The rods can only tell you that there's light or there's no light. I'm looking at the clock. Let's finish up rods and cones and then let's take our break. So other things, rods are low acuity. Cones are high acuity. Low acuity means you don't detect much detail. If you had a book in front of your face, you couldn't read it. You could tell that there's like a book in front of you, but you couldn't tell at all what it says on it. High acuity means you can make out, resolve fine detail. It's like not only is that a book, you know exactly what it's about and what the font is and everything else. So it's like how much detail you can detect. Rods will tell you there's something there. You're not going to walk into a tree, but it's not going to tell you the texture on the bark or something. Other things, rods are on the periphery of the retina. That was not correct. Meaning they're off to the sides. When you're looking at, and then let me just say, as opposed to cones, these are in the center of the retina. There's this little spot in the middle called the fovea centralis where they're particularly concentrated. So if you're looking straight at something, the image falls on your cones, which is this place of acuity, high acuity, kind of most detail you can get. When things are off on the periphery, off to the side of your retina, you can see them, but you can't see the detail. Like, right? So like if everybody like look straight ahead, but put your hands off to the side so you can see their hands, but they're off on the side. you can tell that there's hands there. Wiggle your finger. You still can't even tell how many fingers you got. You just know that there's a hand there. There's something there, but you don't know any detail. That's because the rods are the things that are giving you information about the periphery. Where if you bring your hands right in front of your face, you actually all of a sudden get this real nice detail. You know how many fingers you got, you know the wrinkles on your skin, everything. One of the really weird factoids about our eyes, even though we always think of our eyes being awesome, if you compare them to the the camera on a cell phone, they're horrible. Your eyes would be the equivalent of like a 1 megapixel camera. You know, what's exactly right in front of your face, you can make out with high resolution and a lot of detail. But as soon as you get out of right in front of where the image is landing on this center of the retina, this fovea where the cones are, you lose all that detail. So every time you look, whatever you're looking at is in sharp detail, but everything off to the side, when you look away from it, whatever residual bit of information you have from that, you don't see the detail anymore. So it's kind of wild. Any questions about rods and cones? We are going to come back to them and put them back in the context of the retina here in a few moments. But any questions before we take a break?
Does the light just move across them?
So the light, we're going to see the light has to, it's pretty non-intuitive. The light actually has to move through them to get to where they're actually detecting the light. But because they're transparent, it works. And we're going to see, especially in the fovea, where you're trying to really minimize the distortion and have a high acuity, you're going to have all the other cells that we're going to talk about kind of bent off to the side. So to kind of thin out, make sure that the amount of distortion that the light has to go through to get to the cones is minimal, so you don't distort that much. I don't know if, does that answer your question?
Yeah, thank you.
All right, so let us return, let's see, it's 210, let's say 250. So yeah, we'll finish the eye and I'll think about how much we can actually think to cover with the other stuff and do some labby stuff. I will see ya. All right, so we've talked about rods and cones. And now we're back and now we're looking at the rods and cones in the context of the retina. Catching up, pretending like I never forgot to record that. So the rods and cones feed into the next layer of the retina, which is the bipolar cells. And then you can see the nuclei of those when we look at this retina under the microscope in a few moments. Those then feed into the next layer, which are the ganglion cells. The ganglion cells, those are the ones that have the axons that are going to come out the back of the eyeball. So what are they ultimately going to be creating here? What is this big grouping of axons that are all coming out of these ganglion cells?
Optic nerve.
That's the optic nerve. Also known as cranial nerve number. Quick.
Number 2.
3.
Number 2.
2.
Number 2. So A number of people kind of messed this up on the warm-up from last week. Cranial nerve 2, optic nerve is sensory. That's bringing in visual information from the eye to the brain. Cranial nerve 3, oculomotor, that is efferent, taking messages from the brain to the little muscles to control the movement of the eyeball. So you can also see why you have the blind spot, which you have rods and cones all along here, but wherever this big group of axons is leaving, there can't be any rods or cones. So this is going to become the blind spot or the optic disc here. So there are other cells you do not need to know about them as much. There are also horizontal cells that are connecting up in between in here. There are amacrine cells. So I'll say also, let me just. God darn it. So you have so many neurons and dendrites and axons and stuff in the retina. For what you need to know to recognize in your lab work, the nuclei of the rods and cones are apparent as a layer of nuclei. Then you have basically the axons and dendrites we call the plexiform layer. Then there's going to be another layer of nuclei for all these bipolar cells. Then there's going to be another plexiform layer, kind of the interconnections. Then another layer of nuclei, which are the ganglion cells. And the ganglion cells don't stain as darkly, but you'll still be able to see them. Maybe it's even worth, let me go into it right now. Hold on 1 second here. Pictures, photographs, histology. Here we go. All right, so I am going to share my screen here. There it is. So this is a picture of a retina here. Like this is where the light would be coming in. This layer here is actually the ganglion cells. This layer here is actually the bipolar cells. This layer here are the nuclei of the rods and cones. If you get further back, this is actually the sclera back here. Let me see if the next one looks better. Hold on. This looks better. Okay, this again is still a retina. Let me clear my annotations here. So this part here, you can kind of tell it looks like dense connective tissue. What is this going to be? Assuming the light's coming in this way, what is this part of the eye going to be? Somebody?
Sclera.
That's the sclera. This is the dense connective tissue of the sclera. You know, then we get to the choroid. Then there's this little pigmented layer. That's this kind of really thin, dark layer. These are the rods and cones. So this part here, this is kind of rod and cone, the part that looks like a rod or a cone. These are the nuclei of the rods and cones. And this is that plexiform layer. This is going to be that layer of interconnections between the rods and cones. And then this next layer of nuclei are going to be the nuclei from what cells?
The bipolar cells?
Totally. Heck. Well, that's not great, is it? Let me make that black. So there's the bipolar cell nuclei. Over here, we had the rods, cones, nuclei, nuclear layer. And then that brings us up to here. What are, what are these cells here?
Ganglion.
Yeah, those are the ganglion cells there. So
Good question. So the bipolar cells, is that the plexiform layer, or is the plexiform the bipolar and the ganglion?
it's the thing in between. So plexiform layer just means the layer of interconnections. So there's a plexiform layer between the rods and cones and the bipolar. That's this kind of light zone. Let me get back to my, let me get rid of that. Hold on. Spotlight. What does spotlight do? Oh, what the heck? Can you see that? Yep. So David Eggert is pointing out the choroid layer. David Eggert is pointing out the layer of outer segments of the rods and cones. This is the nuclei of the rods and cones. This is the nuclei of the bipolar cells. So this area here in between is a plexiform layer. There's another plexiform layer in between the bipolar and the ganglion cells. So does that make sense?
Yeah, thank you.
What would we find normally here where my little David Eggert pointer is now?
The vitreous humor.
Yeah, that's where we'd find the vitreous humor. Correct. Check. Okay, I'm getting the hang of this. You can also see that it's a little non-intuitive, but the light has to go through the ganglion cells, through the bipolar cells, through the bodies to actually get to where the phototransduction happens. But that's just the way of it. And like I said, when you get to the fovea centralis, you actually have the other cells kind of folded to the side of it to kind of thin out the distortion. In terms of how the eyes are connected into the rest of the brain, normally we're talking about left side of the body is connected to the right brain and the right side of the body is connected to the left side of the brain. But with the eyes, it's the field of view. Like the left side of your field of view goes to the right side of your brain, and the right side of your field of view goes to the left side of your brain. But both eyes see both fields of view. So both eyes send half of their field of view to one side or the other side of the brain. So let me just draw that, it'll be easier. So this kind of our last piece of talking about vision is going to be, here is your head and your eyeballs, your ears, and your brain, right and left hemispheres of your brain. And both eyes kind of are looking out and see kind of an overlap of pretty much the slightly different, but pretty much the same view of the world. However, the light coming in, like if we think about half, the light coming in from this side of the world is coming onto this half of the retina, on this eye as well as on this eye. The light coming in from the other side is coming in on the other side of the retina for the two eyes. So both eyes have a view of this side of the world. Both eyes have a view of that side of the world. And it's the side of the world that's going to get crossed over on the brain. So this eye is going to send information about the other side of the world to this hemisphere. This eye is also sending information about that side of the world to that hemisphere. Similarly, they're both sending information to the brain about the other side of the world. So this is where you get what you've already seen. What did we call that place where the optic nerve enters the brain?
Optic chiasma.
Exactly. Gosh. Optic chiasma, sometimes called optic chiasm. It's from Greek letter chi, which looks like an X. So that's where you get that kind of X kind of thing where the optic nerve enters into the brain there. It's because each eye is sending half of its optic nerve into one hemisphere and half of its optic nerve into the opposite hemisphere. So that's why it has to make that X-like structure there. And what benefit do we get by having two eyes looking at like the same part of the world?
You get like a broader image or a wider image.
So you don't really, because it's actually narrower. If you actually, in fact, if we look at, you know, like a human, both eyes kind of overlapping, you know, there's not, it's a pretty narrow little sector that they're actually able to see compared to the whole world. If you look at animals like a mouse, where their eyes are on either side of their head, each eye doesn't overlap. So you end up getting almost like 360 degrees of view. So this mouse, you know, you can't really sneak up behind it because wherever you are, it actually can see. Here, if you're a person, it's really hard to sneak up. It's pretty easy to sneak up behind me here because I don't have any visual reception at all behind my head. So what am I gaining by having two eyes both on the front of my face?
More information about the same eye.
Say it again.
Would you have more information about what you're looking at?
And it is more information. And what kind of information? Do you have a...
I think that's where my knowledge stops.
So if you close one eye and you quickly try to touch something and figure out where it is, you basically, it gives you depth perception. I would, after, you know, when you're on your own, spend some time really thinking about what the world looks like when you have one eye closed versus both eyes open. It's a big difference for most people. You have other ways to detect depth, like just like your perspective, things that look bigger or smaller if they're closer or farther, but it gives you a lot of information about depth. Like classic, like if I can, here. Like here's a coyote. He's got his eyes on the front of his face here. Most predators have eyes on the front of their face, and there's some prey, and they want to be able to just get it. And so you want to be able to know exactly where is this thing I want to pounce on, exactly how far is it, what do I need to do to just leap up and nab it and get dinner. Whereas if you are the mouse, you are worried way more about having like 360-degree surveillance to make sure nothing's about to sneak up on you. So in general, things that are predatory tend to, if you think about a hawk or an owl, those are things that, or a cat, they tend to have, if you've never seen one before, they have like their eyes in the front of their face. So that's going to give them depth perception. So they're more likely to be able to pounce and land exactly where they want to be. So that's that. We will leave that there. Any questions about vision before we leave it? Okay. Where's the time? Time is okay. So we're just gonna spend another maybe 10 minutes talking about the chemoreception, and then we'll move into more classic lab kind of stuff. Chemoreception, again, the ability to recognize just different molecules. The stimulus for chemoreception is just a particular kind of molecule. So we can start with gustation, which we usually think of as taste. And in general, when you think something tastes good, it's more than just gustation. Gustation is very limited in what it actually is detecting. There's sweet, there's salty, there's bitter, there's sour. Some people put umami in here. So this is looking what's dissolved in your saliva. What molecules are dissolved in your saliva if you're detecting sweet?
Glucose?
Yeah, glucose or fructose or simple sugars. It's one source, simple carbohydrates in general. Salty, obviously, salts like, electrolytes. Bitter tends to be alkaloids. Alkaloids are a class of molecules typically made by plants, often for defensive purposes. They can be poisonous. If you are chewing on something and you detect the alkaloids, if it's bitter, it means you better kind of pay attention, make sure this isn't toxic. Not everything that's bitter or alkaloids are going to kill you. Some of them are great. Caffeine is an alkaloid, and I'm really happy to put that in my body in the morning. But in general, if something is bitter, that's your nervous system saying, oh, presence of molecules you want to pay attention to. Sweet, your presence of molecules you want to pay attention to because those are giving you energy. So you really, bitter, there's often what do I think about this? Sweet is usually like, give me more. Sour, what is sour detecting?
Low pH.
Exactly. This is going to just basically be low pH or acid. Umami is another one that's actually detecting usually, so these amino acids like glutamate. So like monosodium glutamate, MSG, which is its own kind of unique, kind of yummy flavor, is the kind of quintessential example of umami. So all of these tastes are actually the presence of certain kinds of molecules dissolved in your saliva. The receptor that actually can detect these is the taste bud. So let's talk about the taste bud. Sounds like your little pal to go out to a restaurant with or something. The taste bud. So there's going to be some kind of, in fact, on your tongue, what kind of epithelium lines your tongue?
Stratified squamous.
Exactly. Very good. So we got stratified squamous epithelium. And there's going to be like kind of a little pore, a little hole. And then we've got this structure, the taste bud. It looks kind of like maybe onion shaped or something. It's like a variety of cells that are all kind of clustered together here. There's going to be 3 different kinds of cells, so we'll do them one at a time. These green ones here are they can be called taste cells or gustatory cells. They have these little like, they call them taste hairs. They're little cilia coming off the top. These have receptors. Receptors are basically things in the cell membrane that can recognize and chemically bind onto particular molecules. So receptors that recognize, specific molecules. If this was for sweet, it would be detecting simple carbohydrates like glucose. If this was a receptor for bitter, it would be detecting molecules that are alkaloids. So these gustatory cells basically have these receptors that can recognize and bind. There's actually this chemical binding. And then that actually creates a voltage change in the cell, which then is going to send an electrical signal off to your brain saying that, oh yes, so like here's some glucose, let's say. Glucose is floating in. It binds the receptor. That activates this to send a message to your brain. So the sensory transduction is happening by these gustatory cells that can recognize the particular molecule and then have a electrical response that sends a message to the brain. There are other cells that just kind of help support this whole thing, and they're called supporting cells. And then these taste buds wear out pretty quick, right? You're in this place where there's all sorts of food rubbing around and heat and hot and this and that. So these are constantly wearing down. They're only going to last a few weeks at most. So you have to constantly replace and regenerate them. So there are these little stem cells that are at the base. We call them basal cells. And these are basically stem cells, meaning they can divide, and when they divide, one daughter cell kind of stays behind to be the next stem cell. The other daughter cell starts differentiating and replaces one of the other cells that's wearing out. So these are the cells that are constantly regenerating the taste buds. So it's, even though the cells are wearing out, being at the front line there in your mouth, you constantly have new, recruitment, new cells coming up from behind. So that is your taste bud. Any one particular taste bud can actually respond to more than one taste. So now we're going to look at where do we find the taste buds? Where do we find like 80% of your taste buds? You should know this.
On your tongue.
Exactly, on your tongue. That one was not a trick question. So on the tongue, try to do this. I do my best to do kind of Rolling Stones thing here. All right, so this is supposed to be somebody's tongue. This is coming out of their mouth. And on the tongue, if you've ever looked at somebody's tongue, you know that it's kind of furry looking. The little things that give your tongue texture are called papilla. And there are 3 kinds of papilla that you need to know. So first I'm going to talk, no, I don't want that so thick. First we'll talk about the filiform papilla. They're basically just all over the place. These are the things that make your tongue look furry. If you look at somebody's tongue and it looks kind of furry, it doesn't look smooth. So they're lightly keratinized. That same protein that makes your skin tough, it's what makes your tongue slightly white. When you look at your tongue, it's kind of whitish. It's because these little filiform papilla have some keratin to make them tougher. These do not have taste buds. These are just to kind of give your tongue a little bit of texture and so it can grip a little more. There are 2 other papillae, though, that do have taste buds. It's going to be the fungiform papillae. These do have taste buds. These are just kind of located around. They look kind of like dots. After we're done with this stuff, you should go find, either if you're sheltering in place with other people, you can ask them to open their mouth and say, ah. If you're sheltering in place on your own, just like look in the mirror and you can like see them on yourself. But you can just see these 2 different kinds of papillae on your tongue there. The filiform that are just kind of the whitish fur versus the fungiform, which are more like these bigger dots. And those dots you can know now have taste buds on them. There's one other kind of papilla. They can either be called circumvolate papilla, or sometimes they're just called valate papilla. These also have taste buds. These are big, and they make kind of like, you only have like about 12 of them, and they're kind of making this kind of V-like shape on the back of your tongue. A lot of times people have never seen these. They're really, once you see them, you can't unsee them. They're huge. These big honking knobs on the back of your tongue. Again, if we were in lab, we would have people, and depending on the person, sometimes they're hard to see. Like I can show, like some people stick out their tongue, like and these circumvolate papillae are kind of behind. So you're kind of looking in. It's like, I don't see them. It's because they're kind of hidden. Other people stick out their tongue and it's kind of more like that. And then it's like, oh my God, there are these big honking knobs right back there. So when you look at any one particular person, depending on how their tongue kind of bends as they stick it out, you might or might not see them as well. If you have like a tongue depressor or something to push it down. But if you, again, if we were in lab, we would make sure nobody would leave the class without having seen these if you haven't seen them, because they're dramatic. And normally you don't even realize that you've been walking around with them all your life on the back of your tongue there. The circumvolate papillae have more of kind of like, they're called circumvolate because there's kind of like a little moat that kind of goes around them. We have some really nice slides of these circumvolate papillae, and the taste buds are actually located kind of on the little crevasses. So like this would be a valate papilla from the top, I mean from the side. This would be kind of just the top of your tongue. And these little things, these are the taste buds. In fact, let me go and share. Yes. Okay. This is a valate or circumvolate papilla. Again, this is from our, this is from our collection at school, which you'll never get to see. But alas, wait, why is this acting weird? Oh, I see what's going on. Sorry about that. I am being spazzy here in my control. All right, so this is kind of the, wait, annotate, draw, come on. So I'm gonna share my screen a little different because this is acting weird. Why is this doing this? Sorry about this. It's being a pain in my butt. Okay. Okay, now I think we got it. So now I'm going to annotate, and we're going to put my annotator. So this here, this is kind of the surface of the tongue. This is that stratified squamous epithelium that I forget who, but somebody was mentioning. This is that big honking papilla, big knob. It also has kind of stratified squamous papilla on it, stratified squamous epithelium. It's got connective tissue inside here. And then along the side, you can see these little deals. These are the taste buds. So this is at lower magnification. So the next slide, or the next picture, is the same slide, but at a higher magnification. Let me clear my drawings. So here you can see a beautiful version of a taste bud. This is, you know, again, you're never going to be asked to recognize a taste cell versus a supporting cell. But what you are going to be asked to recognize is a taste bud. And basically, this is the classic looking taste bud. You can see that little pore, the place where those little hairs would come out there. And here we see a beautiful view of stratified squamous epithelium. So you should definitely be able to recognize a taste bud. Just kind of putting it out there. That could end up on a quiz or a test or something, or a warm up, or who knows. So taste bud. If we go back. So taste buds, 80% on the tongue. On those papillae, but then they're on the cheeks, inside your cheeks or down your throat. What cranial nerves carry the special sense of taste?
The facial nerve or the vagus nerve.
So facial is cranial nerve number
7.
Number 7.
Yep, you guess well. Cranial nerve 7, or we should say like 7. Facial. So cranial nerve 10, the vagus, actually does carry some taste. It's a little bit from the very bottom of the throat, like on the epiglottis. Facial nerve is doing like the anterior 2/3 of the tongue. And what's doing the posterior third of the tongue?
The glossal pharyngeal.
Yep, glossopharyngeal, which is cranial nerve number
9.
Uh-huh. Glossopharyngeal. That's the kind of stuff that could end up on an exam as well. What are the special, which nerves are bringing in the special sense of taste or something like that?
What about 12?
12? No, 12 is just motor. If you remember, like, some say Mary Money, but my brother says big books matter more. That number 12 is purely motor. There's no sensory information coming up on cranial nerve 12. So you want to be careful remembering that kind of stuff there. For your sense of smell, it's the same basic idea. I'm going to do it super briefly. So here's your nasal cavity. Here's the cribriform plate of which bone?
Ethmoid.
Exactly. Here's going to be the olfactory bulbs that are going into the brain, the olfactory bulb and the olfactory tracts. And basically you have the same kind of structure like the taste bud, except it's kind of spread out in a whole epithelial sheet. You're going to have olfactory cells that actually have little axons that synapse up in this bulb. This is CN1. CN1 is just the axon of these little olfactory cells. These are cells that have receptors for different kinds of molecules. There's also going to be supporting cells. There's also going to be basal cells to help replace things. But the olfactory cells are the things that are doing the transduction. When their receptor recognizes a particular shaped molecule, it sends a message which goes up to the brain and you have the experience of a smell. But again, the experience of a particular smell is just your perception. The actual stimulus is a molecule with a certain shape. So I can show this to you pretty easy. Hold on one second here. We go into, hold on one second. bleep, 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 bleep. Um, okay, so I'm trying to make it bigger. Okay, I'm going to share this even though it's going to be Dinkazoid. Um, I don't know if you, I don't know, understand why it's not actually zooming for me, but it's not. So this is kind of showing different molecules. It's dinky, but basically different shaped molecules have different experiences. If you have a molecule that looks like this, your experience is, oh, I'm smelling lemon. If you have a molecule that looks like this, that binds the receptors in those olfactory cells, you have the experience, oh, I'm smelling jasmine. You know, if you have this molecule up, you're sniffing, bringing the air into your nose, letting it bind the receptor, it binds this molecule, you're like, ooh, it smells like pee. Yes, this one has the perceived experience of smelling urine. You know, this dude here is peppermint. So when you have a particular experience of smell, the reality is it's your sense of olfaction telling you that actually there is a particular kind of molecule present in the air. Does that make sense? It's like your little chemistry detector set. But rather than telling you, oh, that is a, you know, I don't know, aldehyde or this or that, or it'll just say like, oh, that smells kind of like butterscotchy or something. That's your experience. So it's kind of fascinating. It's cool when you think about what it's actually telling you. And smell is really, really primitive. Like if you were like some bacteria in the primordial soup at the beginning of life, you still got to know like what is in your environment, right? There's going to be, oh, yummy things over here, or maybe kind of noxious stuff over here that you're trying to get away from. So even if you are just some little bacteria in the world, you still want to detect what kind of molecules are floating around. Do I want to like tumble towards something that is maybe a source of food? Do I want to avoid something that might be aversive? So it's this really core experience, core sense. Whether you're a bacterium or whether you're a person, you still need to have a readout on what chemicals are floating around in the milieu. So olfaction is the only sense that has these pathways up into the brain that don't go through the thalamus. If you remember, the thalamus was kind of this relay station where all of the senses came into the thalamus and then got distributed into the cerebrum. But olfaction doesn't need to synapse. What the heck? I do not know what I just did there. So there are some olfactory pathways that do go through the thalamus, but there's also a lot of other olfactory pathways that go directly into different parts of like limbic parts of the brain and stuff that don't pass through the thalamus. So it has a very different kind of look in terms of how it's wired into the brain compared to every other sense. So that's kind of cool. So I am going to leave that there. Any questions about anything we've talked about? About smell, about taste. Okay. So I am going to take you all into, let me make sure I've got it lined up here. Yeah. I'm going to share. All right, so this, what you're looking at here is the Canvas page. One of the new things that I put on here, if you go into modules, I put cadaver dissection videos. So this is under nervous system resources. So there's this group in the University of Wisconsin that actually has these really nice cadaver dissection videos. And I was looking through them, and like, for instance, the brachial plexus, which we've been talking about, they have it really done well on here. You know, these guys are doing regional, you know, kind of studies. So like when they go into a part of the body, they'll say, oh, here are the blood vessels, here are the nerves, this and that. So we have to kind of move around to find the particular things we're looking for. But like, so here I said, like, you know, there is a very nice dissection of the brachial plexus on this video from the University of Wisconsin. You know, here is the URL, and it says start at time 3:30 to see a nice view of the brachial plexus and the median and lateral cords leading to the ulnar and musculocutaneous nerves. Start at time 6 minutes to see the posterior cord and the radial and axillary nerves. So I could just, you know, run it for you right now, and you can see how this goes. So I'm going to go to three minutes. So, you know, this, and it's actually kind of cool. You know, they're starting with just this pro section as part of, it's kind of a little disturbing if you think about it, but move a little farther. They're kind of showing the process of skinning the arm. And, you know, we're going to come back here when we are talking about the veins. Like, here's a cephalic vein. We're going to look at this in more detail when we do cardiovascular. But for right now, what we're going to do is we're going to 3 minutes.
Both of these cutaneous veins travel with some cutaneous nerves. The nerve that travels with the cephalic vein is the lateral antebrachial cutaneous.
Right. Remember, we don't need to know all of these.
And the nerve that travels with the basilar vein.
Can you guys hear the narration on this?
Yeah.
Also
okay.
A branch of the brachial plexus.
Okay, so now he's going to go up to the brachial plexus. You know, the axilla, right? It's kind of, here we go.
The axillary fossa, or classically known to most people as the armpit, is the area that's bounded by the pectoralis major muscle anteriorly. You can see the pec major making the anterior border. Laterally, the humerus.
And you should recognize what he just uncovered there.
And ribs.
And all these muscles should sound familiar.
And teres major muscles form the boundaries of the axillary fossa. When you begin this dissection, the first thing you're going to notice is that there's a lot of fat and fascia filling the axillary fossa.
So one thing, remember that we are going to have a final that is cumulative. So like when he's talking about, oh, and there's latissimus dorsi and teres major and pectoralis major, you know, this is great for you to make sure, like, oh yeah, that's where they are. That's what those are. That's what the names are. So make sure from now until the last day of the class that you use opportunities like this to refresh your memory. Like I'm showing this mainly so you can see the brachial plexus, but it's awesome that he's talking about all the muscles that are making up the little axillary fossa there.
This is that there's a lot of fat and fascia filling the axillary fossa. So it helps to have some sort of a sequential way of going through the dissection. We've obviously cleared out all of the fat in this preparation, and what I want to do is show you a nice systematic way of approaching the dissection. First thing you want to do is find the M. This M is sort of a classic landmark in the brachial plexus dissection. You can see here that the M has a lateral branch known as the musculocutaneous nerve, a contribution to the median nerve, another contribution to the median nerve, and then a medial branch, the ulnar nerve. Once you have this M cleared up and visible, you can then use the M to find all the other branches of the brachial plexus. Begin with the medial branch, the ulnar nerve, and follow it back until it forms the medial cord. You can then look for all the branches that are coming off the medial cord, and there are generally 3 branches off the.
So there's like a bunch of nerves off of this plexus. You don't got to know. He's going to go into nerves that we don't, I'm going to fast forward a little bit here. And now what he's going to do is he's going to go behind. Remember, the lateral cord and the medial cord are from the anterior divisions. And now all of those trunks have, remember the 3 trunks all had posterior divisions that all joined together to become that posterior cord, which is going to give rise to the radial nerve and the axillary nerve.
Branches. You will expose the nerves that are behind the axillary artery that are all branches of the posterior cord. The 2 major branches off the posterior cord are these large nerves right here, the radial nerve, which continues down into the arm and forearm, and the axillary nerve, which dives into the back of the axillary fossa to supply the teres minor and the deltoid muscle. These 2 major branches are the large branches of the posterior cord. So this would be the posterior cord. There are 3 other branches, smaller branches of the posterior cord, known as the upper, middle, and...
All right, so now we're getting into stuff again you don't need. But so this is one thing I'm going to do my best over the coming, like at this point, month and a half. We still got a month and a half of being online. I'm going to do my best to kind of give you directed places where you can actually start feeling more familiar with kind of this kind of dissection where things are moving. Like that PAL, that practice anatomy, has some nice, nice dissections and pictures and stuff, but it's really different compared to like what he's doing, where you're actually moving things and like, let's move this aside and seeing what's underneath it and this and that. So just to kind of give you a sense of what is possible, even if we're not going to ever have a cadaver for the rest of the semester.
I don't know if it's just my Canvas, but that section does not pop up on mine.
Oh, maybe I didn't share it yet. Let me take a look. Hold on. That is really good to hear. Give me 1 second here. Turn on. Oh, I need to get out of this. Go back to here. Modules. Fucking hell. It's because somebody forgot to publish it. All right. Okay, sorry about that. So it should appear now. If you look, I just hit the little button. Again, I am still.
Yeah, it shows up now.
I see it on my end.
Okay. All right. So again, it's going to be partly depending on like how much energy and time. I mean, it's not a sob story, but it's been overwhelming. So I drop in a lot of balls, but I'm going to do my best to like for the cardiovascular, there is some really nice stuff that we usually show on the cadavers with the blood vessels that I would love for you to be able to see. So I'll find some of these with directed viewing for the cardiovascular and things like that. Same thing for the digestive system. So, we'll have some experience of that. The other thing, if you haven't, let me see if I can make this work. Give me 1 second here. Okay. Come on. Oh, fuck, document. Okay. All right, so I am trying to figure out why this is not doing what I want it to do. Oh, here we go. So I was trying, I'm sorry, I was trying to get, next time I will show you this complete anatomy. For whatever reason, it's getting weird on me. But I am going to, I will show you what it can do next time. And I'll also, again, once I'm done with our class, I am going to go send an email to the dean and see if I can get money so everybody can. Yeah, I mean, I don't want to actually share my screen right now because it's not going to be. Oh, why? Oh, I know what's going on. Hold on. Okay, no, I figured it out. I had my iPad was going to my Bluetooth keyboard. Okay, so I'm going to try something to see if this works. Hold on one second. I'm going to go a new share. I'm going to try. Okay, hold on. Oh, I know what I got to do. I got to go. Give me one more second. Oh, I had this working before. Okay, I can't make my iPad share with you guys. Oh, there's one other. Yeah, so I will do this for you next time. But it's a model that gives you really nice control over the whole body, being able to rotate it, zoom in, zoom out, add layers, take away layers. And even though it's virtual, it gives you a much better actual sense of 3D interrelationships than any of the pictures or other kind of simpler models give you. So Thursday, we'll come back to that on Thursday. Let me see. For lab stuff, you know, I would, if we go back, let me go back into share screen with here under this virtual histology lab, which you do all have. If you go into there, and we go into the slide box, I think it's under nervous tissue. You know, again, for lab stuff, oh maybe, oh, they don't. For some reason, I thought they had, dagnabbit, I thought they had the senses, but I don't see them. All right, so, oh, here, organs of special sense. So here, like, so you can go in here and you can think about the eye. You should be able to look at this and you should be able to say, oh my God, there is got to be the optic nerve coming out the back. You know, this is the rods and cones of the retina with those, you know, it's kind of harder to make out, but you can see those layers of the nuclei. The sclera, the cornea, you know, this has got to be the lens. This is going to be the ciliary bodies that have the little muscles that can adjust the focus of the lens. This is probably actually this thing here. If I annotate. This thing, this is probably actually the iris that actually just kind of folded back. Normally that wouldn't be touching the lens like that. But, you know, so I would say, again, just probably it's not going to be useful for you to have me keep talking to you. And I also realized what I need to do is, like, I left I wasn't thinking this was going to end up being permanent. So like your lab manual and things like that, I'm sure we can leverage that in useful ways, but I have all that material still in my office at school. So probably we're gonna have to go do a whole bunch more videos for the physiology labs now that we know that we're not coming back. So I'm probably gonna be on campus later this week to do all that. So I'm gonna pick up more gear and hopefully create more resources for you to use in lab. But I would say for today, you know, go see if you can just feel comfortable with recognizing the retina under the microscope and the different layers of the retina, as well as the bigger picture of the eye. You should definitely recognize taste buds. However, you will not have to recognize the olfactory epithelium. In fact, maybe I should write this out specifically. Under microscope, recognize retina, overall eyeball, taste buds. You know, in a skin, I'm going to just say in a skin model, you should recognize Meissner's corpuscles. Capital because it's a proper name, Meissner's. Again, Meissner's corpuscles are also called tactile corpuscles, Pacinian corpuscles. Pacinian corpuscles are also called lamellated. You know, maybe if it's driving me crazy. I didn't prep enough. I thought I had an easy access, nice picture of the skin, but I don't seem to have one right now. Although if we're in, let me just go into, images.google and skin. Histology model, maybe, yeah. Yeah, so excuse me. I will put a picture up. I'll find one. I'm going to stop talking and instead I'm going to find some nice pictures and I'll create a little thing for you to look at and recognize the skin receptors on. I'll put in some slides of actual microscope slides. You will not be responsible for recognizing the Meissner's or Pacinians under a real microscope. So just to kind of give you more information so you know what the expectations are. Text. General senses will not be tested on actual microscope slides. Kind of two reasons for that. One is the Meissner corpuscles are, you can see them, but they're really tricky. And Pacinian corpuscles are also, we don't find, in fact, the Pacinian corpuscle slides we have aren't even in the skin. For whatever reason, they show up in your pancreas, and we actually have slides of Pacinian corpuscles located in your pancreas. I don't even think anybody knows why they're there. We just know they're there, and it's a good place to nab one to make a microscope slide of it. So general senses, however, your retina and taste buds, and once we're going to get to the ear is the cochlea as well, is something that you could find under a microscope, or in this case, a picture of a microscope slide, photomicrograph. So that's what I would say. Can I review what we've been talking about? If you have somebody who wants to stick their tongue out at you and go, ah, and try to really flatten out the back of your mouth or your tongue, so I would say it's another thing. If you have somebody who's game, try to see and identify filiform, fungiform, and valate papilla on an actual human. And again, this has to be somebody that you're like sheltering in place with, because they're really small. You're not going to see them from 6 feet away. So that is something we would normally do, is actually see these things. So I think that's it. Let's call it a day. I will, as soon as I log out, I'm going to create something in like study guides. On the module for today, I'll have things to help you like for lab stuff, and we'll see you all on Thursday. Unless there's any other questions or anything. All right.
Professor.
Yes, Chris.
Just wanted to say I appreciate the effort you put into these lectures. I know online is rough right now.
Oh, no, thank you. Yeah, no, it's rough. It's been rough.
You're doing a good job.
Cool. Well, thank you very much. I do appreciate hearing that. So, and I am doing my best to try to make sure that this class isn't like a total loss for you guys, because I know that EAST is an important part of what you need to kind of go forward. I mean, especially what's going on right now in the world. Like, I mean, I'm sure it's probably even like a little frustrating to still be in the process of like learning all this stuff instead of just being out there. Although I have to say, my friends who are out there on the front lines, it sounds kind of scary, because I literally have friends who are like 3D printing out things to make face masks. Like one of my friends, like, so his wife could have her whole, her group at Children's Hospital actually would have face masks when they're working with people, so they didn't get infected themselves. It's really not pretty out there right now. It's been pretty disappointing watching the realities of what things are really like out there. And it's also kind of inspiring seeing people working really hard to make the best of what they got. So I'm doing my best to make sure you're all gonna end the semester minimally feeling like you've got like kind of a solid foundation, even if it wasn't as ideal as it would have been if we we're able to all be in class and do it the normal way. So that's my intention, and we'll see. I'm sticking to it. So I'll see you all on Thursday.
Thank you, Professor.
Thank you.
Thank you.
Bye-bye.
Thank you.
Thank you so much.
Thank you.
I gotta get the fuck out of here now.